Good afternoon, dear colleagues. We today continue our cycle of lectures, our webinars. Now it's videos you can find on YouTube channel. In future, I'm planning make a live webinars, and we will choose some platform, and I will give you source of this platform, and I will arrange meeting, and we can discuss every question what you will need. I will be very thankful if you will find ability to provide me your needs. You can leave it on my private links in Facebook, in LinkedIn, or you can leave your questions on the this recording of webinar, and I will discuss it in future with you. Several words about myself, who today I will be thankful who today will find ability, he will watch this video and maybe he will ask interesting question too. My name is Galina Cordero. I'm qualified person of pharmacovigilance, head of pharmacovigilance department, clinical expert at GVP.Pharmac. I'm medical doctor too, have medical education, master degree, and scientific research experience. I have own patterns and articles, and have been written books for medical students too. Why it's interesting exactly for me, pharmacovigilance? I can tell that pharmacovigilance found me in one day, and I loved it from all my heart, because it's more than science, more than activity. It's combining regulatory knowledge, clinical knowledge, and clinical pharmacology knowledge with some changed accent with it easily. What I can say about it, that it's very interesting, and because it's combining not just pharmacovigilance knowledge, but and knowledge pharmacovigilance clinical trials. You should know GVP, GCP, and GXP. And today we will talk about types and sources of safety data. This lecture, why it's very actual, because adverse events, adverse drug reaction, case reports can happen in your practical life, in daily life, and you should know what to do with it. Where you can find case reports, adverse event, adverse drug reaction reports. It can be sources or company reports of adverse events generated during the entire life cycle of the medicinal product, which includes clinical development and post-market period, post-authorization period. It means that you should know again, you should have and GCP knowledge, GVP knowledge, plus another source is health agency databases. It's FHIR's database of United States, Vigilance from Europe, VGBase, Uppsala, and it can be databases, for example, for any regulatory authorities. Now, this database you can get access if you work in a pharmaceutical company in pharmacovigilance. For example, in Ukraine, you can get this access too. And you can use this data together with regulatory authority in order to find a signal and work in signal management in future, and then work with risk if it become risks, and then if this become risk, you should generate and thinking about development, research, minimization measures too. When we're talking about sources of database, it can be studies, and we will talk about today precisely about and preclinical and clinical studies, because it can be source of safety data concerning on drugs, medicines, on full life cycle. Because clinical trials, when we're talking about clinical trials, it can be previously registrations, means that it can be before registration of products. It can be post-authorization clinical trials, and it can be source of safety data too. First of all, first source is non-clinical studies. Second, it's clinical trials, including research in unapproved population or indicators. Observational studies, real-world data, registries, systematic review and meta-analysis, and you should remember about Cochrane database, where you can find a lot of meta-analysis. When we were talking about studies, you can find information concerning on clinical trials and clinical trials Gov site, and use this information in your practical activities. I just would like to pay attention, be careful with meta-analysis, because some of meta-analysis has aim of comparing and from not comparing for reflecting efficacy. Another, for example, can show for example safety issues, and you can use just meta-analysis, for example, who use just efficacy or just safety, and compare it inside, not comparing, for example, meta-analysis with information concerning on with analysis of safety with another one meta-analysis which reflect efficacy of drugs. When we're talking about systematic reviews, now you can find it in Cochrane, and you can find in articles. So many articles you can find right now. You can use database, for example, like Embase database of Elsevier or MBCO. It's the database use it a lot and very frequently for literature searches, European Medicine Agency, and you can use it too. Outcomes research studies, you can include safety data to comparative effectiveness research, comparative safety researches, and health economics analysis too. When we're talking about scientific literature as a source of safety data, we can use, as I said before, it's in base. We include a lot literature source, or for example, it's a very useful database, but you should pay for it, for this database, or for example charge free is PubMed database, but when we compare it to database, I pay attention that Embase include more literature articles and more useful, for example, in your practical activities, but you should pay for it. They have different trees of indexes, and I will talk about comparing this database in separate literature monitoring webinar. When we're talking about scientific literature as a source of exactly safety data, we pay attention on published scientific literature or reports from abstracts, including information presented of scientific meetings, or even unpublished manuscript. And there is, for example, one source is research gates, where you can find even unpublished dates. Use it in your daily activities. Active surveillance system as a source of data, safety data, or Sentinel system or Sentinel sites too. Market access, market research as product usage data and drug use information, a patient support program. This patient support program can conduct product manager pharmaceutical company or together product manager and medical advisors, and pharmacovigilance specialists should be involved in this process of collecting safety data too. Healthcare related data, it's a source of safety data, include administrative and claim data from health insurers, clinical data, including vital signs and laboratory test results, electronic health records, electronic medical records, hospital data, and Medicare data. Too much information, too many sources you can use from internet, but be very careful because you should identify source of this information and remember about validation of this individual case safety report. We remember to live or not live criteria. I will repeat now. It's identified reporter, it's identified patient, it's information concerning on adverse reaction, it's information concerning on exact drugs, suspicious drugs in frames of suspicion of appearance of adverse reaction. What kind of source you can use from internet? It's marketing authorization holder sponsored websites, it's internet search engines, it's patient forums, it's social media channels, but again be careful with indication of source of verification source of this information. Other concerning on internet, why I would like pay attention, because you can meet situation when pharmacovigilance can use as a black instrument of competitive fighting between companies, and as a party there are cases of it, and open source describe it. It's why again you should identify source and get this ability to contact the source of information concerning safety information, concerning on adverse reaction, and for further working with it. Other sources you can use too for working with adverse reaction were indicated. This information, arising from licensing partners, other sponsor or academic institution, research networks. Product quality investigations, information from regulatory authorities, information obtained from VRLs, ad hoc reports generated by company field teams and medical requests of information regarding potential benefits of risk. I got many questions privately concerning on that how work with drug discovery cycle, because as we know pharmacovigilance coming not just for clinical trials. We will now talk later about it. When we're talking about drug discovery cycle, first of all, it's compound collection, because now in this process involved artificial intellect with searching new formula and interesting result you can find in open source concerning on certain molecules of antibiotics, anti-tuberculosis drugs, compounds collection. Why it's important? Because, for example, and connected with pharmacovigilance, because, for example, as you know, L form and right form isomers has different toxicity, and L form has higher toxicity comparing with right form. And when we remember thalidomide, we remember different isomers, and one of them, as you remember, got this teratogenic effect, and you should remember it. It's why now very important has knowledge not just for clinical, as I all time repeated in my webinars, clinical knowledge, clinical experience, medical knowledge, but there are very actual specialists and profession. It's pharmacovigilance specialists in R&D, research development. Why? Because they should have this group of specialists should have and clinical experience, clinical knowledge, clinical pharmacology knowledge, medical knowledge from medicine, and research development knowledge, and medical chemistry and chemistry knowledge, analytical chemistry knowledge, and biological chemistry knowledge. It's combining knowledge. It's why this kind of this group of specialists now very needed in pharmaceutical company easy. You see that compound collection, chemical synthesis, and going to primary assays, high throw put in vitro, then and it's non-clinical trials. Then it should check in secondary assays, contents, increase bioavailability, toxicity, metabolism. It's because drugs should be work in organism, in live organism. It's a question about bioavailability, toxicity, metabolism. We will talk about these processes when we will talk about non-clinical trials. Lead components and CAR and structure characterization of protein ligand complex. It's all, as you see, connected in drug discovery cycle, and you should know it too. I got question about clinical trials phase in my privately questions, and we will talk about it more precisely today. When we're talking about pharmacovigilance, if before it was information that pharmacovigilance starting from clinical trials, now if we see it now that it's drug discovery, find right molecules, mouse prototype of molecules, work with it. Because on this stage of drug discovery and preclinical trials, you meet situation with exploratory toxicology, and this investigation in vitro screens, for example in silica screens mutagenicity, cytotoxicity, and hepatotoxicity. Single and repeat dose range finding studies in 2 species. And when we talk in investigation in animals, you should know and model which using animal model. For example, some non-clinical trials conducted just on rabbits, some can conduct just on mice. It depends on from what kind of system will you check it in these clinical trials, what kind of drug on what kind of system its influence, and you should know it. Plus we're talking about model of disease, for example diabetic mellitus model, or another for example model which you should restore in clinical make this model on this kind of animal. You should have this knowledge, and we will talk about in different webinars. Now we will talk about general knowledge in frame of types of source, in frame of clinical trial, in frame of exactly you can use for detecting adverse events, because clinical trials, it's a very deep topic, and pharmacovigilance in clinical trials, it's more deep problem which we will learn further. What learned clinical trials phase 1? It's safety, it's pharmacokinetics, its problems tolerability, and what's interesting in this phase, it's that in this phase conducted this clinical trials conducted on healthy humans, volunteers, and for example, when we're talking about pharmacovigilance in this stage, for example, antihypertensive drugs. When we're talking about antihypertensive drugs, main effect of this group of drugs is decreasing pressure, arterial pressure, and for example, in 5 patients, clinical investigator noted decrease of pressure, for example, more than 20 measure, and you see its effect of main drug, investigated drugs, or its adverse event. Again, why pharmacovigilance specialist in clinical trials should get this clinical knowledge, because primarily this assessment making investigator in clinical trial, but later it's doing exactly pharmacovigilance specialist in clinical trials. In clinical trials phase 2, investigated safety, efficacy in is not big group of patients, a few patients. When we're talking about limitation of clinical trials, it's one of limitation. It's not big amount of patients in clinical trials, and very difficult to extrapolate these data on real population because of amount of patients. Plus, we remember including and excluding criteria for clinical trials, and when we're talking about for extrapolation these dates from clinical trials, this population, real population, has many diseases and many conditions, plus pregnant ladies, plus its pediatric population, its special population with disease of liver or kidney, hepatic disease, kidney disease, or for example, a patient with cancers too. Clinical trials phase 3, its safety, efficacy, many patients too, and post-American surveillance as ongoing pharmacovigilance, broad clinical population. I don't pay attention in this when I'm talking about this slide about amounts exact, because my accent is on role of pharmacovigilance in clinical trials. What I would like to make accent on different things. It's regulatory GLP toxicology, which can be source of safety data and you should, when you find it in preclinical trials, you should expect that this can be in clinical trials too. It's why when, for example, it's very dangerous, it can be even reason of stopping clinical trials. And for example, when we remember on some groups of drugs, when you see that in preclinical, for example, in mice, you see malformation in mice, and you see that this drug, for example, anti-epileptic drugs, you can expect that this malformation can happen in kids from pregnant who took these drugs. And we see, for example, this situation very close in valproate drugs, when teratogenic effect in kids from pregnant women who gave birth for kids, and these kids can have these teratogenics problems. It's why you should pay attention on it in non-clinical, preclinical phase, because clinical trials, in majority cases, pregnancy is excluding criteria, and when this drug go to post-authorization period on markets and pharmaceutical markets, and it can become even indication, we can get very big problems afterwards. And its problems is valproate repeated after teratogenic effects. It's why now there are some government programs in countries, and I think it's big benefit, because these drugs are rejected in pregnant women too. It's why we're talking about toxicology, and teratogenicity, about fertility, influence on fertility too. And you should pay attention not just on women and men, because sometimes it can influence on exactly on fertility of men too. Regulatory toxicology, its safety, pharmacology, genotoxicity, 28-day repeat dose toxicology in two spaces, 3 to 12 chronic toxicology in two spaces, reproductive toxicology, 24 months carcinogenicity in two spaces too. Why I won't show research studies, because you should know it when you involve it in pharmacovigilance in clinical trials. You should know type of study. There are two main type of studies. It's observational, descriptive, and experimental, interventional, analytical. Plus, I can add, when we're talking about pharmacovigilance, it can be epidemiology studies, and it can extrapolate on pharmacovigilance, pharmacoepidemiological study too, according GVP. This knowledge you should have too. Observational, descriptive research studies divided on cohort, case control, case report can be a case series. Plus, there are case report can be experimental, intervention, analytical. It's uncontrolled clinical studies, clinical research studies. When we're talking about cohort or case control studies, design of study, it's cross-sectional prevalence, it's longitudinal incidence, and retrospective can be prospective studies. When we're talking about experimental, interventional, analytical, it can be uncontrolled and controlled, and this when we're talking about design of these studies, parallel crossover can be what you can use its superiority, equivalence, and not into inferiority, and can be masked or often open labeled study. About a clinical study and, for example, blinded and unblinded study, and it's question very now important, actual, when we're talking about clinical study, because unblinding we will talk about later when we will talk about standard operation procedure, unblinding of clinical studies, because before it, pharmacovigilance specialists, clinical investigators, nobody will know before unblinded what drugs can be source and reason of adverse reaction. For example, so it can be placebo or investigation drugs, or it can be comparator or drugs investigated, which can investigated in these clinical trials. And this procedure should must be for unblinding and algorithm of activities should be right, and because if it's not be done, clinical trials can get big problem in future. And drug discovery, we going now more precisely. We were talking about drug discovery. Typically this involved highly parallelized processes for making new compounds and testing them in higher throughput screens. From this, a certain number of hits will be obtained, and this will be whittled down by first analysis into set of leads. Preclinical trials, this includes in vitro and silico tested of the compounds to identify the best members of a series to take into clinical trials. This is also where the first stages of safety assessment are undertaken via toxicity tested in animals. If a drug shows promise in preclinical trials, a pharmaceutical company can request permission from the, for example, regulatory authority, from FDA, from HRA, from EMA, to begin tested in humans, known as first in man or film trials. This is called investigation new drug (IND) application. In Europe, the European Medicine Agency (EMA) equivalent is an investigational medicinal product dossier (IMPD). Clinical trials phase 1. You remember that it's conducted on healthy volunteers. Phase 1 clinical trials are concerned primarily with establishing how a drug is absorbed, distributed, metabolized, and excreted by the human body, a study known as pharmacokinetics (PK) studies. The dosage range of a new drug is determined but administering increasingly larger doses to one or more groups of subjects who are closely monitored for harmful side effects. The goal is to learn the maximum tolerated dose that does not produce unacceptable side effects. Phase 2 clinical trials are designed to answer the question: does drug X improve disease efficacy? How is this drug effective and in patient? It's not healthy volunteers, it's patient with disease. Subjects in phase 2 clinical trials may benefit from their participation in the receive an active treatment. Most phase 2 clinical studies are randomized, with subject assigned randomly by chance and not by choice to receive the experimental drug, a standard treatment or placebo, harmless inactive substance. Since larger numbers of patients receive a treatment in phase 2 clinical trials, there is a greater chance to observe and compile information or potential side effects. Phase 3 clinical trials are conducted in multiple centers with hundreds or thousands of patients for whom the drug is intended. Testing on large patient population allows continuous generation of data on the drug safety and efficacy. As in phase 2, most phase 3 clinical trials are randomized and blinded. A drug in this phase can be studied for several years. As you see, and assuming it makes some resume, types of sources of data, the safety of medicines is under evaluation throughout the drug development cycle. This process starts before humans are exposed and continues during the clinical development and post-marketing phases. Broadly, the safety of a medicine is tested in 4 phases, each of which produces different types of data: preclinical animal studies, healthy human volunteer studies phase 1, clinical trials phase 2 and phase 3, and post-marketing surveillance phase 4. Also, there is a natural sequence defined by the above order. The phases are not entirely distinct. Sometimes new preclinical studies are undertaken for authorized products, and clinical trials are increasingly becoming important post-marketing. Systematic review and meta-analysis are important tools for bringing together data from multiple studies. Although they have usually been applied in the assessment of efficacy, their use for safety purposes is increasing. Which direct question concerning on non-clinical and clinical development of drug? In using powerful query functionality, we can ask ICH2 yes questionnaire. We can ask a variety of direct and indirect safety related questions. What information are looking for and what questions need answered to ensure that a drug will be safe? Is there toxicity in the liver, heart, cellular metabolism, or reproductive organs? Is the drug carcinogenic or genotoxic or mutagenic? What is the therapeutic dose level for these drugs? Is it close to an unsafe dose level? Has this compound been tested before, perhaps for a different disease? Is the pathway completely predictable? Are there any drug-drug interaction? And is the drug metabolized in the body? Indirect question concerning on non-clinical and clinical development of drug. Is there any relationship between known toxins and current drug? Is there any relationship between toxic pathway and the drug in question? Potential common factors could include proteins, tissues, genes, substructure, distribution. Are there any toxicity associated biomarkers related to the drugs? Is an animal viable without a knockout gene? that is implicated in a pathway. And drug similarity questions. Do dissimilar compounds have known toxins? Are there toxicology studies on similar compounds? Similarity could be structural, 2D or 3D, mechanistic, and based on end effect. The flexibility of ICH2 enables you to answer this question precisely, tailoring queries to extract exactly the information you require and then combine results into desired format. Assessing safety during preclinical trials. During preclinical trials, the critical question pharmaceutical developers seek to answer is whether the new drug is safe to be tested in humans, which is also the primary concern of regulatory agencies. The safety assessments start early, and as candidates advance from discovery to clinical trials, more extensive tests have to be performed in vitro and silico. The rapidly growing discipline of computational toxicology and in vivo to gain a better understanding for their pharmacodynamic (PD) and pharmacokinetics (PK) behavior and establish their pharmacologic safety and toxicity profile. Preclinical trials are the final hurdle prior to clinical trials, and only 12% of the candidates advance to phase 1 clinical trials. From this point, the success rate increases at each clinical phase, with 17 at phase 1, 27 at phase 2, 58 at phase 3, and 82 at the registration phase. An average drug discovery and preclinical development takes 3 to 6 years and account for 30 of cost per drug. And you see source of this information, it's cost of development and new drug briefing, Taft Center for the Study of Drug Development. Preclinical studies are usually conducted in rodents, rabbit, mouse, rat, and dogs or primates, monkeys. They aim to establish the dosage level below which toxicity is not observed and to identify the organs adversely affected by higher doses. The most important potential effects studied are major organ toxicity, acute and chronic toxicity, carcinogenicity, mutagenicity, able to induce genetic mutation, and teratogenicity, producing physical defect in the embryo. Remember valproates, remember thalidomide. Preclinical studies. Adverse drug reaction may not be specific to particular species. When studies in animals demonstrate major toxicity, further drug development is usually precluded and the level of toxicity in humans remains unknown. When no major toxicity has been demonstrated in animals, development can proceed into humans, but some adverse reaction appears to be specific to humans. The multisystem oculomotor syndrome caused by beta blocker Proctor law. It's generally accepted that thalidomide teratogenicity was not predictable from animal studies. Generally, the predictive value of preclinical studies for human toxicity is not more than moderate, and discussion continue about relevance of this study to human use. Overall, they provide only limited reassurance the use in humans will be acceptable safe. Early identification of potential safety toxicity issues. This early identification of potential safety issues is crucial to optimizing investment R&D and avoided failures later in the drug development process. You see how important this early identification of potential safety toxicity. And this it's like examples from open issues. You can see how it's look like that investigated dose, for example, toxicity and another characteristic. Human volunteer studies phase 1. Human volunteer studies phase 1 for most medicine. The first human exposure takes place in healthy volunteers, but cytotoxic drugs used to treat cancer are an exception. Participants are very closely monitored with the clinical supervision, and resuscitation equipment must be immediately to hand. The purposes are to establish a possible dosage regimen, investigate how the drug is handled by the body, and what the effects are on a variety of standard parameters: pulse and blood pressure, EKG, hematology. Assuming the drug appears to have no major untoward effects, it can then be studied in clinical trials that include patients with the target diseases. Healthy volunteers of phase 1 studies have generally had a good safety record over a long period of time, but occasionally major adverse reactions do occur. In 2006, all 6 of the first humans treated with the monoclonal antibody TGN-1412 in London rapidly developed multi-organ failure. The incident was investigated in details by the United Kingdom Regulatory Authority, who concluded that the reaction were an unexpected biological effect. A further unexpected and serious incident occurred in France in 2016. You see, it took some time when administration of escalation doses of BIA 10-24-27, a fatty acid amide hydrolase inhibitor, to healthy volunteers led to 1 death and 3 cases of serious neurological damage in healthy volunteers. In the instance, about 80 people had previously been given the drug without similar effect being observed. Clinical trials phase 2 and 3 studies. Clinical trials are usually designed to study both efficacy and safety, but efficacy is usually the primary endpoint. The design of such trials incorporate various features to minimize bias, such as randomization to treatment groups, blinding of subjects and observers, investigators to treatment allocation, and validated measurement instruments. Initially, very small phase 2 trials are conducted. These tend to be focused on efficacy and other requirements. Larger phase 3 trials are then conducted and form the key element of the safety database prior to marketing. All adverse events occurring in patients after exposure to the drug and comparator, which can be a placebo or an alternative active drug too, are systematically recorded. In order to minimize measurement bias, it is usual to blind all study participants, patients and clinicians, which is known as double blinding, to the treatments given, or, if it's not possible, to blind those who are involved in assessing the outcomes, a form of single adverse events that occur at significantly higher rates of the medicinal products of interest than one comparators. Usually, the data from all pre-licensing trials are pooled in a global safety analysis for presentation to regulatory authorities. Clinical trials will identify most common adverse reactions, but often have important limitations, including selection of patients, those at greatest risk of adverse reaction, or those who have not responded to similar medicines are often excluded. Study size. The numbers of patients studied is generally not enough to identify rare but serious adverse reactions. The artificial conditions. Patients are likely to be more closely monitored than in real life. Measurement of surrogate markers of effect rather than hard endpoints. The duration of follow-up is usually short, weeks or months rather than years. 1 to 10 clinical investigator brochure for safety assessment. We know documents. Investigator brochure is Bible for clinical investigator. One top 10 pharmaceutical company has provided access, and every company has research for writing this brochure. And in clinical for investigators, it's very important previously to conduct by monitors specific trainings. It's like line, first of all, it's a pharmacovigilance specialist conduct training for monitoring, and then monitors teams conduct trials for investigators. It's very important to try give and all general knowledge and specific knowledge concerning specific products, because investigators should pay attention and have all information which reflected in investigator brochure easy. Limitation of clinical trials. It's a limited size. Let's assume this topic. It's a limitation of clinical trials, limited size, short duration, narrow population, narrow set of indicators, and concomitant medication which are absent in clinical trials. Good case management follow-up procedure, prioritized by value of the case, highest priority for serious unlabeled followed by serious labeled, then non-serious unlabeled. Adverse events. Non-serious labels should not be followed up if criteria are met, as treat special issue and events that might lead to label changing as higher priority. For priority cases, obtain as much information as possible during the initial contact. The extent of follow-up details solicited should be driven by the seriousness and expectedness. Use CIOM's triage algorithms. For serious unlabeled case, follow-up until the long-term outcomes is known. If reporter does not cooperate with telephone follow-up, send written reminders. Acknowledgement letters should be sent to suppliers of follow-up. It do not encourage rechallenge. Factor affecting spontaneous reports. We talking about spontaneous report as a potential source of adverse reaction. Spontaneous reports it's more often and frequently using in pharmacovigilance as a method, and it can be you can first of all conduct training for pharmaceutical company, and every specialist, every worker of pharmaceutical company can be source of adverse events and spontaneous reports. Everybody from your company should know you as a QPPV. You should be 24 hours, 7 days online, and they should know what kind of information these workers specialists should provide you, know and be trained by you not less than one time per year. If we're talking about medical doctors, healthcare professionals, what you can do, you can provide information about yourself as a QPPV and form for collecting adverse reaction in your site, and it will be very useful for every patient, medical doctor, nurses, healthcare professional, pharmacists to fill out these forms, and you will get information. Why it's important? Because you can need more information and request follow-up information, and this work will need your time, will need your knowledge and skills, and it's one of the main part of pharmacovigilance. It's working with source of information, with patients, with nurses, with medical doctors, with pharmacists. And what kind of factors affect spontaneous reports? Its volume of use, the duration on market, its severity of reaction, its label, its status, its new molecular entities, its manufacturer, its publicity, its calendar year, awareness of reporting. Everybody should be aware of adverse reaction. Medical doctors should teach and train patients before administration of drug, and medical doctors should be trained by clinical pharmacologists in the hospital, and attend specific courses as pharmacovigilance. And what I see right now, that pharmaceutical university and medical university should create some specific courses for it, because this knowledge is sometimes very specific, and medical doctors need this knowledge, but sometimes medical doctors and pharmacists are so busy with routines that have no time for it. But it will save life patients, and we remember that patient is first for everybody in this chain, and for pharmaceutical industries, and for medical doctors, hospitals, for nurses, healthcare professional, pharmacists, from all community who are responsible for health of patients. Patient safety is first. Limitation of spontaneous reports. It's adverse event recognition. What I see right now, the majority of medical doctors has they get scared report about it, because some of them thinking about medication errors. But I think, and I see that for example, medical doctors from United States look on it different way, and they report more than European medical doctors. And but we everybody from this chain should all time 24 hours 7 days get this knowledge, and this continuity and of knowledge received should be should be endless, because you see that it's very important. Estimated exposure data and quality of report. Sometimes medical doctor, pharmacist, and nurses can say, for example, "Oh, I'm sorry, I don't have enough time for fill out form," but when medical doctor, pharmacist, nurse will have, for example, will have some, convenience form, and reminded that he can fill out it on, for example, site form, and report not just to regulatory authority but for pharmaceutical company, because case management, case assessment, signal management, risk management, and in the end it's risk minimization measure development conducted, and assessment. These processes making pharmaceutical company now maybe more often than even medical doctors do. Strength of spontaneous reports: broad exposure, cost-effective, and signal generation. And when we will talking about signal management, I will describe you and reflect you situation where exact spontaneous reports make source of signal management, signal in signal management and signal detection of this, and it's a real serious situation. Represents everyday use. Regulatory safety reporting requirements. International standard in general. Serious unexpected suspected adverse reactions. It's unblind reportable clinical trials cases. Is suspected fatality written cases 7 calendar days, and all other reportable serious suspected reaction 15 calendar days. Right now, European Union, if you live in the European Union, you should report a non-serious adverse reaction, but you have time not less than 90 days, and for 15 calendars again, it's for serious expected and unexpected reaction. You have to report 15 calendar days. National requirements beyond accepted international standards. Example: all France, all study-related serious adverse reaction. Ireland, all serious adverse reaction irrespective of labeling and blind and occurring in domestic centers. United States, all fatality written serious adverse reaction events irrespective of causality within 7 days. For very specific drugs genetically engineered. Finland, India, Norway, Slovakia, Switzerland, all domestic size, irrespective of causality. Dear colleagues, thank you for your attention. I will be very appreciative of your questions, of your needs. If we can discuss, if we can make workshops for topics, if you, for example, will need some specific information, I will continue conduct webinars. It will be again 2 branches. One branch will include question concerning on clinical pharmacovigilance, drug safety problem, and it will be general and specific field. For example, it can be general, for example, pharmacovigilance pediatric population or drug-drug interaction, and then it will be specific, for example, pharmacovigilance in psychiatry, pharmacovigilance, gastrointestinal, it's diseases, gastroenterology, pharmacovigilance, and cardiology. Why? Because I would like to provide general information, then assume it and talk about specific field, clinical field. It can be useful in clinical trials, for specialists in pharmaceutical industry, and for investigators, and for clinical investigators who involved in clinical trials, and for medical doctors, physicians, and different profession, for example, cardiology, gastroenterology, because problems like polypragmasia, where patient administrate too many drugs, it's very sometimes dangerous, and it should medical doctor know all diseases, concomitant diseases, all drugs which were administered by different previously medical specialists, medical doctors from different field. And another branch, it's regulatory. It's for pharmaceutical industry, question concerning on how to build pharmacovigilance system, main mistakes and critical pharmacovigilance processes. It's concerning on good pharmacovigilance GVP and how it's implemented, main mistakes, main errors, and how we work it. It's question concerning audits and how, for example, pass successful inspection. It's I have experience in this field and I have conducted more than 40 webinars previously, and I will assume I update this knowledge and will provide to you, and you maybe some topic will be interested to you and it's not included, not to be provided, you will not find, for example, in list of this program. I think that I will represent all program to you, and you can find is this topic interesting to you, this topic is interesting to you, or you will need some new topics, or for example some new knowledge, because for example when I monitor this knowledge, this information, I've looking for this information, it's my job, pharmacovigilance, and this information you can consider will be actual for you today. It can related on in classes for specific class specific effects of drugs, on your risks and how work with risk, how minimize these risks. I'm sorry for so long speech, and I how explain. I'm very thankful that you have that you found this time for watching this video, and I will be appreciated to see you in another hour meeting, and I will arrange a live meeting, and I will make announce of it, and I will be very thankful to see you in this meeting. We can discuss online, and for me it's a very interesting experience too, because previously it's very hot discussion, because your knowledge, my knowledge, your experience, new experience, we discussion sometimes we can even argue, but true, and this real information, real situation, we can discuss real cases, from clinical practice, how work with this clinical practice cases. It's very actual for medical doctors and situation. For example, now I prepare webinars. It will be live webinars, main finding during inspections, and I think it will be hot discussion too, because for example, if regulatory authority will find even one critical issue for critical processes, it can be reason of stopping circulation all drugs, all medicine, or pharmaceutical companies. Why again our two main tasks of pharmaceutical industries: one is patient first, it's providing of effective pharmacovigilance monitoring of efficacy and safety of drug, working with signal management, working with risk minimization program and measures, and control efficacy of these measures. And another aim, another purpose of pharmacovigilance system is inspection and readiness, and be ready in any time to accept pharmacovigilance inspection, and not just pharmacovigilance, because from my previously experience, it's very important. You can be involved as a QPPV pharmacovigilance specialist to inspection, for example, a GCP, good clinical practice, or for example GMP, good manufacturing practice inspection, because for example, when we talk about it in my specific webinars, you will see real situation, and very important to organize very effective communication and collaboration with, for example, between QPPV and QP in company, and from it will depend success too, because QPPV and QP it's two main person who responsible for efficacy, for safety, for quality of medicine or pharmaceutical company. Thank you very much for your attention. I wish you a nice time and see you later on my first webinars.